Hi everyone. It's always such a pleasure to be able to talk to people about our research, and I'm really proud to have Eduardo here today, one of our PhD students in our lab, to talk about what he's doing. So we are going to talk about winter barley primarily, and ultimately we are getting into the same comparison that the ISA on-farm trials are trying to make. We're trying to compare this directly to cereal rye for weed suppression. And Jim, you did set me up very, very well. So, you're talking about the amount of cereal rye that you need for weed suppression. And across several years of a multi-state trial, we've been able to determine that at 3 foot tall cereal rye, you probably have about 3 tons per acre. And that's the ideal amount of biomass that you need to suppress water hemp and palmer. So yeah, that's the cereal rye number, right? But the question is it less for winter barley? So I want to tell you more about why we think it might be less for winter barley. So, I want to thank all of these people, so all the students in the lab, everyone that works with us. I just stand here and talk about the research. It's really these guys that do all the hard work. So I really appreciate all the effort that the students put into our lab. So today, what we're going to talk about, I'm going to start just an overview of integrated weed management and why we need it. And as part of that, I'll give you an herbicide resistance update. And then we'll talk about winter barley and integrated weed management. I'll talk about allelopathy for a minute, and then I'll turn it over to Eduardo, and he'll finish up the talk for us today. I almost always present this slide because it really sets us up well to tell people why they should care. This is the global cases of herbicide resistance and how they've increased over time. And these are unique resistance cases, so species by active ingredient. Now, this is across the globe, and this is all management systems. It's not just agriculture, but it really shows you how much pressure we put on herbicides to manage our systems, because this is intense selection pressure. It's a probabilities game. The more acres we apply that particular herbicide on, the more likely we're going to select for that one individual plant that has a mechanism to overcome that herbicide. So really, that's what we're looking at here is pressure. And then this is the newest thing that has happened in weed science, I would say, the newest big thing. So we've been watching our post-emergence herbicide options decline. And now we are to the point where we have found the first case of glufosinate or Liberty resistance in water hemp globally. So this is work that's just being published. It's been accepted for publication now, but we were able to work with the University of Illinois, the Pat Tranel lab, to confirm this resistance. So I want to tell you a little bit more about that and how that was found, because this is really your checkoff dollars at work. It's really unfortunate we were able to find this, but I would not be telling you about this today if we had not had funding from our checkoff programs. So this was presented at the WSSA, the Weed Science Society of America conference in 2024. Our leads on this were Drs. Young, Bradley, and Norseworthy. So they collected all the populations from all the states, including Illinois and Southern Illinois. And basically, this was, we were collecting escapes at the end of the season in soybeans. We were driving around the countryside. We could find escapes that looked like a suspect resistance population, where you could see a patch in the middle of the field that seemed to be spreading outward. That was what we were looking for, the pattern. So we would knock on the nearest door, try to figure out if we could go into the field, and then we would collect those seeds and screen them. So really, this was funded by USB. And we have been able to confirm glufosinate resistance really early in resistance evolution. So we were surveying and monitoring across multiple states with this project. We used the 1x field rate for a rough screen of these populations just to see where we might have problems. So we screened for dicamba, glufosinate, and 2,4-D. I just want to focus on the glufosinate for a moment, but we have data for all of those for multiple states. We screened when the weeds were 3 to 4 inches tall, and there you see the flats here. There were about 25 plants per flat that were sprayed with these 1x rates, and then we rated them. Now, what I want you to pay attention to here is the yellow and the orange. That indicates a moderate risk of resistance and a high risk of resistance, because I'm going to show you a map in a little bit that's color-coded. So pay attention to the orange especially. So we were able to rank the flats by their probability for resistance. And this is what the Southern Illinois population looked like, one of them, for glufosinate resistance. So it's what you would expect to see, with a little bit of necrosis on the tips of those leaves there. And when you start looking at it, the plants are still green. You just see this little bit of necrosis, and the plants keep growing. So this is the map I want to show you, right? Because it looks like, this looks really bad for me, right? Am I doing a bad job in Southern Illinois helping people manage their water hemp? So we see the orange all across Southern Illinois, right? So that indicates that we may have a problem in multiple counties. Now, when we took these individual accessions back into the greenhouse, we did very specific dose response curves. We sprayed each population with multiple rates of herbicide, and then we tried to figure out what our effective dose was that killed 50% of the population. From all of that work that we did, we could only confirm resistance in Southern Illinois in Franklin County. But you see that we have some other suspect populations. We're probably seeing the early stages of resistance evolution, and our dose response curves just aren't able to capture that yet. So I want to show you this because we need to put more pressures on our weeds besides just herbicides, right? So just to go into that a little bit more, working with Dr. Tranel, again, thank you for the funding from ISA that went to the Tranel Lab, and the United Soybean Board also funded my component of this project. So we looked at these four populations. In Northern Illinois, they had a suspect population from Carroll County, Kankakee County, and McLean County, and then we had our Franklin population in the south. And then we had our two susceptibles, the WS and BRC populations, were what we used to compare these two. So when we took them into actual screens, and I'm gonna explain the graphs that you see here, but we can tell you that these resistant populations compared to our susceptible had a 3.4, up to a 3.4-fold based resistance on survival, just looking at survival alone. And then a 2.8-fold increase based on dry biomass. So this is definitely resistance much above that 1x rate that we're targeting, that we're looking at. And just for reference, the only other amaranthus that's been confirmed to be glufosinate resistant, it came out of North Carolina. And well, they're actually having problems in, I know there have been other states as well now with Palmer. But this particular case had a 2.3-fold resistance. So water hemp is expressing a higher level of resistance than some of the Palmer that we've found lately. So just looking at one of these graphs and what this looks like, when we run our dose response curves, you see along the x-axis at the bottom, the glufosinate ammonium dose. So we have multiple doses along that axis. And then we have our survival along the y-axis, so the number of surviving plants. Now, the point is looking at the difference between these two lines where you see the dotted line coming out horizontally. So that shift to the right that red line shows, that's what indicates resistance. So that is showing a higher LD or ED50 or LD50 rate. So that's our established growth rate. For this, GR50 is the rate that decreases the growth rate by 50%. So we go back and we look at those other graphs for all the populations, and you can see the shift to the right of all those curves compared to the susceptible population. So that's essentially how we confirmed. And then the Tranel Lab did a bunch of molecular work, including whole transcriptome sequencing. And what I want to point out here, the thing that is the most important is this one here. So these are the number of genes that were expressed differentially between these populations. So they only share one expression in common. That indicates, as well as some other data from this, that indicates that we're possibly looking at a couple of different mechanisms here. So this is metabolic resistance. It's not target site. So basically the plants are taking glufosinate and they're breaking down that molecule into something that's not active internally anymore. And there are multiple ways that these populations are doing this potentially. That indicates independent evolution at these multiple spots, right? So this is showing the tendency of water hemp to evolve resistance now. So we know this can happen across the landscape now. Bright spot is that, if you have an amaranthus seed bank and you can effectively control it, then you lose 90% of that seed viability within about 3 years. So this is our justification for really trying to suppress these populations. We're never going to get back to eradication. There's always going to be some seeds that are viable in that field, but we can dramatically reduce the population levels if we can maintain good control for 3 years. So that brings us to integrated weed management and kind of how do we do that? So diversified rotations, including cover crops, are a really great cultural tactic to try to accomplish this. We know with cover crops, here you just see a rolling cereal rye at anthesis there, so talking about the large amounts of biomass that are needed to suppress water hemp in particular. But cover crops are taking the resources and the space that these weeds need, right? So that's the way they're suppressing them. And then with winter barley, Eduardo and I are really focused on the allelopathic potential of winter barley. And allelopathy is a concept, you can kind of think of it as almost like chemical warfare between plants in a way. So one plant produces chemicals that suppress the growth of the plants around it. It can be beneficial if those chemicals stimulate microbes that indirectly stimulate other plants. But what we are looking at is a negative impact of winter barley against the weeds, right? So I want to just point out, you're all familiar with this concept. This plant may not look familiar right now, but it has a compound. It produces a compound. So it was observed that plants that were growing around this plant in Australia were exhibiting bleached leaves and were not growing as quickly or as successfully. So when this plant was looked at a little bit more closely and this compound was discovered, it was eventually used to synthesize Callisto. So you're all familiar with mesotrione as a product, the HPPD herbicide that's used in corn primarily until now with the Viconic soybean system. But you even see the bottle brush flower on the label. So that brings me to Eduardo and what we're trying to do here in the lab. So Eduardo has been working primarily with winter barley and trying to quantify the allelopathic effects of winter barley. And he's compiling a list of all the potential allelochemicals that could be found in winter barley. And it's going to differ by cultivar. We may actually see that some cultivars of winter barley are much, much better than others. And we're actually kind of beginning to see a signature of this in the research that Eduardo is doing. But we have cyanoglucosides, flavonoids, polyamines, phenolic acids and the derivatives, hydroxamic acids, and alkaloids. So this is just a chemical soup of all kinds of things that could potentially suppress weeds. But with that, I'm going to turn it over to Eduardo, and I'm going to let him talk about his research.
Well, thank you, Dr. Gage. Thank you, everyone, for being here today. Let me start introducing myself. I'm Eduardo Lago. I'm a third-year PhD student in the Weed Science Lab at SIU. And as you may or may not have noticed, I have a pretty thick accent because I'm not from here. I'm from Southern Illinois. I'm from Brazil. And it's my third year here working with Dr. Gage. So we're looking at allelopathic. I have two trials I'm going to show you guys today. One is more of a lab work. We're looking at what chemicals are being released by barley into the soil and compared to cereal rye. And we have one trial evaluating the weeds emerged on a field with winter barley. The first trial, we are comparing, the objective of the trial is compare barley and cereal rye and see which one has a higher response on water hemp or Palmer amaranth in this case. We're looking at how does the chemicals produced by these plants can interfere on the growth of Palmer amaranth. So we thought before we started that both plants would inhibit Palmer amaranth, and we had the questions, what if we introduce barley to Palmer prior, and then we look at how is the response to barley from it. Pretty much we had the question, what if the Palmer annoys barley? Is it going to become more aggressive to Palmer? To test that, we planted in the greenhouse barley by itself, barley with palmer, cereal rye, and cereal rye with palmer. We transplant those plants into these boxes. I'm going to show you more pictures of it. It's called the plant box method, where we get a donor plant, a plant that is donating these chemicals to the soil. We move into this box inside of these tubes, and we plant Palmer amaranth seeds on top of the box. This box is filled with agar, which is like a gelatin. Think of like a gelatin inside of a box. And the idea here is let barley diffuse these chemicals into this gelatin and see how Palmer respond to these chemicals. So before I talk about the results over here, that's how we set it up. So barley over here, Palmer in different distance. And that way we can calculate the growth of roots and shoots of Palmer. And we see, that's where it gets really cool, the research, that barley was able to inhibit 45% of the root and 31% for the shoots. That was more than what cereal rye inhibit. So 31 for roots, 14 for shoots. And it did answer our question. Barley, both of them inhibit Palmer amaranth, but barley was able to inhibit more. And our second question, barley that first met with Palmer was able to inhibit even more. Same for cereal rye. So what does that mean? It means these chemicals produced by barley are a potential source for us to use it on the field and inhibit the growth of these weeds on the field. There is previous studies done, but not by us, but other researchers, and they look at these chemicals, that list of chemicals that Dr. Gage presented. That is a compilation from other studies, and these chemicals are responsible. We're going to look closer into them and see the difference between cultivars and how they produce these chemicals. So barley root exudates were able to influence the growth of Palmer amaranth. That was just 5 days enough. 5 days of interaction was enough to reduce its growth. We also see that plants that were previous competing with Palmer amaranth, they were able to inhibit even more of the growth. And we have more research ongoing that one of them we're repeating on the field. We're going to talk about them right now. And we have another research coming up, which is going to be the different cultivars. So the second study is on the field. So we move from the lab, plant barley on the field, and we count the weeds on the field. The objective here is see how much winter barley is able to inhibit the germination of water hemp and giant ragweed. So over here we're working with water hemp instead of Palmer. We didn't think it was a wise idea to introduce Palmer seeds into our fields, and we have a natural population of water hemp. So we are working with our natural population, plus seeds that were introduced to these fields. So we hypothesized here that the presence of barley was going to be able to inhibit the growth of the weeds. And we also want to test here different conditions for this barley. So we had different termination timings. We planted green, or we planted the soybeans into green barley, and then we have later terminations. We also tested a pre-emergent herbicide with it. So that's barley full grown. Soybeans are breaking out of the ground over here. That's 7 days after we plant the soybeans, and that is later on 28 days, I believe. So important dates over here. We had barley was planted on October 15th, or soybean was planted in May 12th of the following year. The termination times we had was 7 days prior to planting the soybeans, at planting, 7 days after planting the soybeans, and we also had a 14 days after planting to compare only for the soybean yield. The field also received an even application of herbicide across all treatments with 21 days. And we counted the weeds 21 and 42 days after planting the soybeans. And at the end of the season, we also look at the soybean yield. This graph over here shows the germination of these two weeds in natural conditions. We have the number of seeds emerged on the field, or on plots we set up. Control plots where we had the weeds over there. We had counted the number of the weeds introduced, so 1,000 seeds from each. And on the x-axis shows the time of the year. So blue line, water hemp, germinating on April, started germinating on April and germinating throughout the whole summer. Giant ragweed had a different response. It starts germinating way earlier on the season. So the first day we counted the seeds or the emerged plants, we already had giant ragweed emerging. And it reaches plateau early in the summer. Also, these blue lines over here shows when we did the termination timings. So the middle one over here is May 12 when we planted, 7 days prior, 7 days later, and then we also have the 14 here for yield. 21 days later, we did the first counts. 42 days later, the planting, we did a second count. And what we found there is so much. We see every single graph I'm going to show over here is going to be the number of the weeds on the left over here and the treatment applied down here. So we saw interaction factor between pre-emergent and termination timing. So whenever we did not have pre-emergent for water hemp at 21 days, we had fewer weed counts later termination. So 7 days terminating the barley, 7 days after we planted soybeans, we had the fewer counts for water hemp. However, when we introduced the pre-emergent, don't matter, the termination timing doesn't even matter anymore. All the plots with pre-emergent had lower counts. As expected, we want our pre-emergents to be working pretty good, right? And barley itself was not a factor that interacted with these other two, but we saw that plots with barley, they were effective on reducing the water hemp on the field. So this graph, this darker line over here is the mean of the counts. Plots with barley barely had any water hemp. For giant ragweed at 21 days, same trend, but termination timing was not an effective factor here, but pre-emergent was, as well as barley. Water hemp at 42 days after planting, pre-emergent was still holding the emergence of these water hemps, and barley as well. For giant ragweed, only barley was able to still reduce this weed on the field. When you look at the yield, so I split over here a graph with no pre-emergent. We're going to see one with pre-emergent afterwards. This, I have 3 termination timings to show here. And our yield is over here, bushels per acre. And if you want to speak science, tons per hectare over here. We see, don't matter the termination timing, if you do not have barley on the field, our yields were lower compared to barley. So termination time did not affect yield at all, but only the presence of barley was able to boost soybean yields. Same for non-pre-emergent. Don't matter the termination timing, so we have 7 days prior, at planting 7 later and 14. Don't matter the termination timing. No barley, lower yields than with barley. With that, we conclude that barley is a potential tool to reduce the weeds on the field. It can increase our yield, as we saw it. It is effective to hold the emergence of this yield. Oh my God! It's effective to hold the emergence of this weed early on the season, so less competition early on the season for soybeans. And barley, it can be an important tool for our integrated weed management. So I'm not saying herbicides will take out of the equation. Herbicide is still important, as we saw for pre's, but cover crop or different cover crops will be another tool for us to use here. Just like you need a hammer on your toolbox, but you also need a wrencher or a screwdriver or anything else. And we also doing future studies here. We're looking at barley and cereal rye side by side. We currently have those plots on the field. We're going to be conducting this trial over this year. And we're going to be giving them the same treatments and see which one does better for soybean yields and counts as well. And I want to thank you all for listening. If you guys want to see this data up close, there's a copy of the presentation, and just scan it and you guys can see it again or understand it better with not a thick accent speaker, you know. Thank you.